And we will get started. Hello, everybody, and welcome to our latest still COVIDing chat. We're here with our awesome friends from Profi Nasal Spray, Alex Trevellos and Dr. Nitin Ghoshi, two of the founding members of Profi that are here today to share with us the data that they have on Profi and Influenza A, of which H5N1, the bird flu that we're all sort of following closely now. That's a type of Influenza A, so this is really relevant information. And then, of course, they'll share with us, regular Profi information for those of you who are just joining us. These guys are a wealth of information about a really cool tool and product we have to keep ourselves safe. So we will hold questions until the end, just like we normally do. And with that, I will turn it over to Alex. Thanks for being with us, Alex.
Thank you, Kara. Let me share my screen so everyone can see the presentation. Looking good?
Looking good.
All right, so wonderful to see everybody today. I see a lot of familiar names and some new ones as well. So before we get going, I just want to thank Kara, Sandy, and really the entire mod team. I see Christina on the call for graciously hosting us this afternoon. There's a ton of work that goes on behind the scenes to coordinate these sessions, and we're very, very grateful for the opportunity. I also want to thank all of you for being here today on a Sunday afternoon and taking time out of your weekends to join us. And also thank you for everything that you do to keep people informed on the risks of respiratory infections and the latest and greatest science. So with that, let's get rolling. So we're going to start with some introductions, a little bit more about who we are, and provide some background information on our company. There's a story behind Profi that will really set the stage, and so we're going to walk everybody through how we came together, and then we're going to do a deep dive into the science behind Profi. If you've been on one of our previous calls, one key difference is that we're going to weave in as part of the discussion, the specific ingredients that are driving different elements of performance into the discussion, and especially when it comes to influenza and the avian flu. And then I have a few product updates in case anybody on the call has used Profi before. Be delighted to share those, and then a special thank you offer at the end of the session, and then we'll of course open things up for Q&A. I just want to remind everybody that this presentation is provided solely for educational purposes and should not be construed as medical advice. Always consult a qualified healthcare professional. So my name is Alex. I'm the CEO of Akita Biosciences and one of the co-founders of the company. We are the company behind Profi Nasal Spray. A little bit about me, I'm a biomedical engineer by training. I spent my entire career in healthcare. Before co-founding Akita Biosciences, I had the opportunity to work in respiratory care, managing a brand of, I would call them specialty devices sold into hospitals. So everything from novel SpO2 sensors that rest on the nasal ala, to devices that help to inflate the cells within your lungs so that you can breathe better and clear mucus, as well as protective hydrogels that help prevent pressure ulcers for individuals on non-invasive ventilation. And so, one of the things that really struck me about, being in that role is just the global burden of respiratory infections and pathogens. And this was before the pandemic. And I always like to remind people that, if you look at the 2019 CDC data on mortality, influenza and pneumonia is a top 10 killer. And then some of the chronic respiratory diseases like COPD and asthma are, I think it's like third or fourth. And when so often we see these problems kind of work in tandem, and people go to the hospital and unfortunately they don't always make it out. And so, I was moved by the magnitude of respiratory infections and diseases, and really wanted to spend more time on products that keep people out of hospitals and keep them healthy instead of, treating them when they're sick. And so that's why I decided to co-found Akita Biosciences with the rest of our amazing team. And so, Dr. Joshi, I'll kick it over to you for an introduction. And if you could speak about Jeff as well, it'd be great.
Thank you, Alex. Hi, everyone. Really nice to meet you. We are looking forward to share, as Alex mentioned, a lot of information about Profi, scientific data, and here to answer your questions. So I am an assistant professor at Harvard Medical School, Brigham and Women's Hospital, and one of the scientific co-founders of Akita Biosciences. We developed Profi in my lab and Dr. Jeffrey Gobb's lab. We started this. Alex will talk more about this story later on, but the idea sort of started pre-COVID, and then when COVID hit, we were working on this platform and decided to spin into a different direction. My lab works mainly on biomaterials and drug delivery, addressing unmet medical needs across a range of different applications. Nasal and inhalation delivery is one of the key focus areas of my lab. We are also interested in developing longer-acting injectables that can address patient compliance issues with chronic conditions. We are interested in developing novel vectors or carriers that can deliver therapeutics and gene therapies across the blood-brain barrier for neurological diseases. I was trained with Dr. Jeffrey Karp, who is one of the scientific co-founders of Akita Biosciences. I did my postdoctoral fellowship with him. So Jeff and I have been working really for almost 10 years now together. And he is the distinguished chair and professor at Brigham and Women's Hospital, Harvard Medical School, and also has an affiliated faculty position at MIT. He is an elected fellow of the National Academy of Inventors, Royal Society of Chemistry, and American Institute of Medical and Biological Engineering's College of Fellows. Jeff's research has been mainly focused around bio-inspired medical problem solving, and the technologies developed in his lab have resulted in almost like 12 companies that have been translating technologies to address unmet medical needs across a range of different applications. So really a prolific inventor, great scientist and mentor. And it's been really a great pleasure to have such a great team together, that co-founded Akita. And we are really looking forward today to talking to you more about the product. Thank you.
Thank you, Nitin. So before we get into the science, I want to just take a step back and ask everybody to picture this. So it's March of 2020, and the world is suddenly gripped by fear and uncertainty. And if you look at the news headlines, you might remember words like lockdown and words like unprecedented. And healthcare workers were really stretched to their limits during that time. Hospitals were becoming overwhelmed and running low on really critical healthcare supplies like masks. And so one of our co-founders, Dr. Jeff Karp, he was actually part of an emergency meeting at a large healthcare system to strategize and to come up with contingency plans. And the question that they were asking is, what are we going to do if we run out of masks? And we didn't know it at the time, but over the subsequent months, it became clear that the SARS-CoV-2 virus is airborne, and it spreads when somebody who's infected, whether they know it or not, coughs, sneezes, gags, or even just breathing and talking, and they release contaminated aerosols into the air. And we learned this by following the science. And we also learned that the nasal cavity is the primary entry point for the virus, and that it targets the ACE2 receptors within your nasal passages. And I think at this point, it's really pretty well established that the nasal cavity is key. It's of course not the only route of delivery. It can be transmitted through the oral cavity, through your eyes, but we know that the nose is really key. So fortunately, before the pandemic, so go back even further several years, and certainly before we created Profi, we were deeply invested in understanding the best ways to deliver therapeutics intranasally. So our research was inspired by knowledge that many of the new molecules and biologics in the academic labs and development were were emerging, and the ideal route of delivery for these therapeutics, particularly CNS drugs, was the nasal cavity. However, these new molecules were significantly different than anything that had previously been delivered intranasally. So I included naloxone as an example, but when you look at naloxone, which is of course a life-saving anti-opioid, opioid reversal medication, it's quite small in the grand scheme of things. It's about 360 Daltons, and it's like, almost by virtue of its size, you can spray it into the nasal cavity, and it'll have some degree of efficacy. However, when you look at the new, drugs on the horizon, they're significantly larger, over 400 times larger than molecules that had been delivered previously in the nasal cavity. And it's not just their size and weight; it's also the shape and the way that they interact with cells that made it quite difficult for the nasal route. And so, give kudos to Jeff and Nitin and the entire team, but they developed a platform that allows us to encapsulate molecules within a hydrogel, which can be sprayed and forms a uniform coating within the nasal cavity. This coating is designed to have a long-lasting contact time, which is really a key difference from other nasal sprays. And so as another example, when you look at a nasal spray like Flonase, which is of course used for seasonal allergies, about 50% of that drug is either absorbed or cleared within about 20 minutes. And by the time you've reached an hour, 80% of it's gone. And so there's a very short window of opportunity for the drug to take effect. So our focus, of course, was on providing a solution that extended the contact time within the nasal lining and to allow molecules to gradually be released for several hours. So we began experimenting, obviously, looking at the science and looking at the platform that had been built. We asked ourselves, can we develop, really tailor our nasal spray in such a way that complements existing solutions like masks and vaccines by shielding the nasal passages to protect against emerging pathogens as well as existing pathogens. So I'm excited to share with you all, yes, we can. The data behind Profi is set to be published, we hope very, very soon. Unfortunately, we don't always control the timelines, but we're in the final stages of publication. And that paper is now titled Towards a Radically Simple Multimodal Nasal Spray for Preventing Respiratory Infections. And what you'll see in the study, as well as in the data slides here, is the formulation is referred to as PCANS, pathogen capture and neutralization nasal spray. So with that, I'll kick it over to Nitin to walk you through the data.
Thank you, Alex. So as Alex mentioned, we started this really before COVID, and when we were trying to develop this platform for the delivery of large molecules across the nasal cavity, one of the things that we accidentally found when we were testing the formulation in mice was this very long residence time in the mouse nasal cavity. So when we looked into the papers, the residence time of the nasal formulations that people had reported previously in the mouse nasal cavity was around 1 to 2 hours. And we're seeing we're seeing the signal from the formulation that we were instilling into the mouse nasal cavity for almost 8 to 10 hours. And then we started digging into that, and I'll talk a little bit about that later on, what's responsible, for such a long residence time. But when we saw that really got us excited to develop this technology for preventing, respiratory infections. But then the question was, how do we, further engineer this platform to really maximize the blockage or the neutralization of different types of pathogens, not just SARS-CoV-2, that can cause different respiratory infections. So here, so really if you break down this platform into different properties, there are really these 5 different angles here. So one is long-lasting property of the formulation, and then the second is its ability to capture droplets and aerosols from the air that carry the pathogen, which are created when someone coughs or sneezes, and its ability to block the transport of these pathogens through it, which is essential to prevent the interaction of these pathogens with the receptors in the nasal cavity, and finally, the ability of the formulation to neutralize these pathogens with a broad spectrum activity, and ultimately the safety of the formulation. So we call this formulation, especially in the paper, we call it PCANS, which really means pathogen capture and neutralizing nasal spray, because it can capture the droplet, capture and block the transport, and then also offer a neutralization ability. So I'll walk you through some of the data that really shows that show all of these properties individually. So the long-lasting effect of the formulation that we observed in the mouse nasal cavity was in this experiment where we tagged the formulation with a fluorescent dye, instilled into the mouse nasal cavity, and we were able to see a very long residence time. And what we have done is really we screened at the beginning, pre-COVID time, when we were trying to screen different components of the formulation that really maximizes the nasal residence time. We found three components to be really, really critical for this property. One is gelin and pectin. One is gelin, another was pectin. Both of them are biopolymers, so these are polymers derived from natural sources, and both of them have property to stick to the mucus layer, so they can bind to the mucus, to the mucin proteins which are present in the mucus layer through electrostatic forces. And previously, people have used this property a lot to really create drug delivery technologies and other type of technologies to enhance the residence time of the formulations which are intended to be used on the mucosal surfaces. But here, gelin does one additional thing, and what it does is it has this unique property of forming a gel in the presence of salt or electrolytes in the physiological fluid, including nasal fluid. So what happens is gelin is a negatively charged polymer, and when there are cations in the physiological fluid or in the surrounding environment, for example, calcium or something else, it can really trigger the gelation of gelin molecules. So they can interact with these ions and can form these hydrogel network structure. And when we studied the mechanical properties of the gelin gel that results from the gelation of the gelin polymer, the mechanical properties are really 100 times higher than the properties of the ungelled gelin. And in this process, what we did was we wanted to ask if gelin was really critical. So we screened different polymers, including gelin, pectin, and several other biopolymers, to see if we get the similar mechanical properties in gelin. And gelin was the only polymer that resulted in that 100-fold increase in the mechanical properties. And later on, we found that was, and I'm going to show you the data later on, but we found that was also really important for the formulation to have a very good physical barrier property. So overall, to summarize here, gelin and pectin have mucoadhesive properties. Gelin, in addition to the mucoadhesive property, also forms a very tight hydrogel network. And then we had this third component here, which is polysorbate 80, which is present in many commercially available nasal formulations. And by the way, when we were screening for different ingredients, one of the filters that we applied there was that all these ingredients should be present in the inactive ingredient database of the FDA, or the generally recognized as safe list of the FDA, and must have been used in commercially available nasal or topically applied products. So all the components that are present in Profi are present in commercially available nasal or topical products, and are present in Profi at levels either lower or similar as compared to the levels which are present in the commercially available products here. So polysorbate 80 in this particular case is also so we screened different amounts of course, these components to really identify the optimal amount. And we made sure that the optimal amount here, we only used it if the optimal amount is really less or similar to the commercially available product. So we found that polysorbate 80 was really critical. So gelin and pectin could lead to, let's say, 2 hours of residence time in the nasal cavity, but adding polysorbate 80 was really the game changer. It really enhanced the nasal residence time from that 2 hours to really 8 hours. And then we digged further into the mechanism, and the way it works is that polysorbate 80 and many other surfactants have this ability to really slow down the cilia, the movement of the cilia, which is responsible for clearing things, especially mucus, in the nasal cavity and other parts of your body. So the movement of the cilia really moves the mucus and clears things from a nasal cavity. What polysorbate 80 does, it slightly reduces its beat frequency to a level where it doesn't cause any toxicity concerns, and we confirm that later on by repeated administration of the product, which I'll show you later on. So the combined effect of gelin, pectin's mucoadhesiveness, along with the gelation of gelin to form a strong layer with a very good mechanical strength, plus the polysorbate 80's ability to reduce the mucociliary clearance, resulted in a long residence time of 8 hours. And we demonstrated this in mice, and we believe that this would also be applicable in humans, because in humans the mucociliary clearance is actually slower compared to mouse anyway. So if something lasts in a mouse nasal cavity for like 8 hours, it has to last in the human nasal cavity for slightly longer. So this is the data that I was talking about earlier. So what we did here was we basically tagged the formulation pecans with a fluorescent dye and instilled it into the mouse nasal cavity. And the control here in this experiment was just the free dye to make sure that that the fluorescent signal that we are getting over a long period of time is not just because of the dye getting into the tissue. So as you can see here, these are the representative nasal cavities from one mouse at each time point. And as you can see here, the signal was observed from the dye signal was observed for up to 8 hours in the mouse nasal cavity that it was instilled with pecans. But in the case of the mouse nasal cavity that was instilled only with the free dye in PBS, the signal was almost gone by 4 hours. And one additional thing I would like to point out here is that by 24 hours, as you can see, the formulation is almost gone, which means that that the reduction in the mucociliary clearance because of PS80 is not permanent. It's only temporary, and the formulation eventually gets cleared, which you really want, so that there's not accumulation of there's no accumulation over time of the formulation as you redose it, and which is very critical to prevent any toxicity concerns. So the next part that we really optimized was the ability of the formulation to capture the respiratory droplets that are laden with pathogens. So when someone coughs or sneezes, they create these droplets and aerosols that contain the respiratory viruses or pathogens. And what happens is in this aerosol and droplets, the droplets are basically, these particles of size 5 to 10 micron or larger, and less than 5 to 10 micron, it's really aerosol. And the particles with size less than 5 to 10 micron can go into the deeper lungs, but 5 to 10 micron particles are larger, mostly deposit in your nasal cavity. And these are the ones that are really, really important for transmitting infections, because your nasal cavity has the maximum number of, entry checkpoint or receptors to which the virus binds. So as you go into the deeper airways, those receptors reduces in number. So the chances of getting infected are way higher from a nasal cavity compared to the lungs or deeper airways. And that's why nasal cavity has been shown to play a really prominent role, and people are trying to really develop technologies that would really prevent the infection from spreading through the nasal cavity. So the goal here was, so when we were looking at what some of the other products that people had developed before or were developing at that time, we saw that a lot of people were interested in either like blocking the virus physically or neutralizing the virus via some chemical interaction or something else, right? But when we thought about the process, the most important thing we really felt is to really capture the respiratory droplets, because the respiratory droplets have a tendency to sometimes bounce off of your nasal cavity surface. So because of the aerodynamic properties, they can deposit in the nasal cavity, but because of the ability to bounce off, they can also just like leave the nasal cavity and go into the deeper airways. So we wanted most of them to be trapped so that the risk, so that you have the minimum number of droplets really going down into your deeper airways. And to do that, what we did was we screened a number of different surfactants, because surfactants can really reduce the overall surface tension of the layer, and that helps in bursting the droplets, as they hit the surface of the coating. So, so to do that, we screened like polysorbate 80, benzalkonium chloride, and tween 20, several other surfactants, and we found that polysorbate 80 was the one that really resulted in the maximum capturing of the respiratory droplets. And when we added that along with gelin and pectin, what we saw is what you see here in this formula, in this example. So in this study, by the way, we use a 3D printed nasal cavity. So this was not done in mouse, because it's very hard to like create the aerosol and then make sure that that goes into the mouse nasal cavity, because it's really tiny. So we used a 3D printed cast of the human nasal cavity with all the intricate anatomical features inside, and then we nebulized a solution that contained virus-like particles. So these are virus-mimicking particles, and these particles are loaded with a dye. And then you used a nebulizer to nebulize them, and then you do this nebulization for I think 10 minutes, and then you take the nasal cavity out, and you see how much dye was trapped within the nasal cavity in case of mucus-coated nasal cavity versus the nasal cavity that was coated with both mucus and pecans. And as you can see from the data on the right, when we had the nasal cavity coated with pecans along with mucus, we doubled the capture of the respiratory droplets, which was very exciting. And this was the first report that really demonstrated anything like this. So as I mentioned, all the previous reports developing other similar nasal formulations were not focusing on this important feature. And we think that the suboptimal efficacy of those products that have been demonstrated in clinical trials or in preclinical models is primarily because of the lack of this important feature. So after you capture these respiratory droplets that are laden with the pathogens, and the droplet really bursts open at near the formulation, the next step in this multi-model approach is really to prevent or to block the transport of the pathogen through the formulation. Because if the pathogen can really get transported, then it's going to bind to the entry checkpoint anyways, and then the whole purpose is defeated. So we screened, again, as I mentioned before, different polymers, including gelin, carbopol, pectin, carrageenan, and xanthan, and several other biopolymers. And we looked at their ability to prevent the transport of different types of viruses, bacteria, and even small molecule dyes, actually. So the way we did this experiment was, it was an in vitro study. You take a strainer, and then you coat the surface with either mucus or with Profi or pecans. And then you add a suspension from the top that contains your pathogen. And then you quantify how much pathogen has been transported through it into the bottom chamber. And as you can see in all of these data, pecans basically prevented the transport of influenza A, SARS-CoV-2, K pneumonia, E. coli, RSV, adenovirus by almost like 99.99%. And when we digged into the mechanism of this, we found that it was really because of gelin. So if you remove gelin, for example, from the formulation, then the blocking property of the formulation just completely disappears. So it's really it's really beautiful that each ingredient here has been really optimized to maximize the efficacy of each step in this multi-model approach. So we have gelin here really contributing towards the effective prevention or effective blocking of the respiratory pathogen to get transported across the formulation. Now, once you have blocked the transport of the pathogen, you don't want the pathogen to be, just being there and leaking out in some way later on. So you want to neutralize it chemically or physically somehow. So for this, we did a very rigorous study, and it's available in the paper online as well, and which is going to come out soon, where we screened alcohols, surfactants, biopolymers, and we looked at influenza A, SARS-CoV-2, and different types of pathogens. So before we mixed all these components together, what we did was we individually screened the capability of these surfactants, alcohols, and biopolymers for each type of pathogen, and we found was that, for example, pectin was really critical to neutralize influenza A. And similarly, BKC, benzalkonium chloride, was very critical for the neutralization of SARS-CoV-2 and for the neutralization of the bacteria. So what I'm trying to say is, if you remove pectin now from the formulation, you lose the ability of the formulation to effectively neutralize influenza A. You'll still see some neutralization because of the other ingredients, but it's really like a synergistic action, that is really happening here, where different components are, playing like the key role to maximize the efficacy for each type of pathogen for the neutralization part. So once then we also confirmed what is the minimum amount of each ingredient that is needed to maximize the neutralization effect. For example, in the case of pectin, we evaluated 4 or 5 different concentrations of pectin to study the neutralization ability against influenza A. And we found that, okay, this is the minimum concentration, that we really need to achieve greater than 99.99% neutralization for influenza A. Similarly, we did for BKC, we studied for SARS-CoV-2, we studied for K pneumonia, we studied for E. coli. And then, because some ingredients here are cationic, some, for example, they have positive charge, some have negative charge. We also wanted to make sure that when you mix them into one formulation, they do not counter the effect of each other, because they might just like bind to each other and result in and then the complete loss of the efficacy, right? So for this, what we found, we did these experiments, where we combined different amounts of different ingredients together, and we found that, okay, we can combine X amount of pectin with Y amount of BKC to really prevent their interaction. And if you increase BKC now or increase pectin beyond that, then it just completely counters the effect of the other component. And finally, the order in which we added these ingredients was very critical as well. If you add one component before the other component, the formulation might not work, because they might just gel together. So there was a rigorous in vitro experimentation that really went into the development of the formulation, especially from the neutralization point of view. And from the data, as you can see here, when we really combined all the formulations together, we were able to see greater than 99.99% neutralization for different types of pathogens, both virus and bacteria. Now, there's a lot of news these days about avian influenza virus, which is H5N2, and the influenza A virus that we studied in our in vitro experiments was H3N1. And we have been thinking a lot recently, how this formulation would really work if for avian influenza virus. So first of all influenza viruses share some surface properties. So the proteins on the surface, which really form the receptor binding domain. So the receptor binding domain is essentially the protein structure on the virus that binds to the things, into the receptors in your body, and through which the virus makes its entry. Sometimes it's also called the virus's spike protein. And the way we neutralize the viruses, or Profi neutralizes the viruses, the way it happens is really by binding of the pectin or BKC to the proteins on the receptor binding domain. And what we have found is that the receptor binding domain of the avian influenza virus H5N2 has way more positively charged proteins, which play a critical role in binding of pectin to the spike proteins on the surface of the virus. So what that means is that the pectin will have propensity to bind to the spike proteins much more strongly in the case of avian influenza virus compared to the H3N1. So what it implies is that if we have seen greater than 99.99% neutralization of influenza virus H3N1 with pectin, we should definitely see that effect with the avian influenza virus H5N2 because of the presence of the proteins on the surface that are more favorable to binding with pectin here. So after we did a lot of rigorous experimentation in vitro and using 3D printed nasal cavity or ex vivo, we then moved to this experiment, which was performed in mice to really demonstrate the prophylactic efficacy of Profi or pecans in vivo. The study was done in collaboration with a world-leading infectious disease expert at Brigham and Women's Hospital. And what we did here was we used we used a mouse model of influenza A virus. In this particular case, this is the mouse-adapted H5N1 strain called PR8. And this is a very, very lethal virus, has a very high virulency. Only 10 particles of the virus are just enough to kill the animal. And we tested the efficacy of Profi in this particular experiment against a 25 times the lethal dose of PR8. So we administered 250 particles of the virus to these animals. And the way we did this was we treated the animals prophylactically with pecans of Profi, and after 30 minutes or 4 hours, we exposed the animals to the virus intranasally as well. And then we monitored these animals over time for their body weight changes, survival. We also looked at the lung viral load on day 2 and day 4, and looked at several other parameters, including cytokine, and cytokine counts, and also the counts of the inflammatory cells in the bronchoalveolar lavage fluid. And when the animals were euthanized, we looked at the lung histology. So as you can see from the data on the left bottom, animals that were treated with only virus, died very quickly, by day 6 or day 8. And then, but compared to that, the animals that were prophylactically treated with pecans survived during the entire experiment. So the 100% survival was something that we saw the first time. So if you look at when we looked at the previous papers, where people have developed similar formulations, people had to dose, people had to do the prophylactic dosing multiple times, and also the survival that was observed was 60 to 70%, which is still pretty good, but not 100%. And when we saw 100% survival in our study, especially for the supra-lethal dose of this virus, this was really, really remarkable and really motivated us to move this forward towards translation. And then when we looked at the lung viral load at day 2 and day 4, you can clearly see that in the animals that were treated with pecans prophylactically, there was a greater than 99.99% reduction in the lung viral load, which was truly remarkable, and also matched with the in vitro data that we had seen before. We also, as I mentioned, we also looked at the cytokine levels. We saw the cytokine levels back to normal. That is similar to the cytokine levels in the healthy animals. Similar to the BAL count, and lung histology showed significantly reduced lung inflammation in the case of a pecans-treated group compared to the only virus-treated animals. So this was really, really the milestone, the greatest milestone for us that really motivated us to move this towards translation. I think that's all right, Alex, on the data. Yeah.
Yeah.
I think there's a question which I can take later on, I think.
Okay.
On the chat. Yeah.
I don't have visibility of the chat, so I'll trust your answer. So we'll look at Q&A at the end of the session. I did just want to share a quote. Is it cheesy? Maybe, but I do think it's relevant here, and that's of course by Steve Jobs. That simple can be harder than complex. You have to work hard to get your thinking clean and to make it simple. And I share that because, often when you're in the development process and when you're innovating, it can be really messy. And as Nitin shared, there were hundreds of experiments and different combinations of ingredients to try to figure out what works, what doesn't, and to establish the right, concentrations of key ingredients. But if you've done a good job, and once you kind of take a step back and get out of the development, ultimately the solution will be really simple, and it's almost like it's the obvious solution. And it's shame you didn't see it on the front end when you started. And I think that's relevant, and I share it because it's truly one of the things that makes Profi so unique, is that we didn't go and try to reinvent the wheel. There's no new drugs or new molecules here that are being sprayed within somebody's nose. every ingredient in Profi, and there's only 6 of them, has been approved for use and is on the FDA's inactive ingredient for nasal use at higher concentrations than what we have in Profi, with the exception being gelin, which has been rigorously studied and we think is very, very safe. But it's not easy for ingredients to make it on the FDA's inactive ingredient database. It takes a lot of toxicology data, in vitro data, animal data, of course, approval of the product, and then really years of post-market surveillance. And when the FDA thinks it's safe, they may include it on the IID. And so again, that's what makes us unique. Everything is on the IID or gelin, which is on GRAS. And I think it's important pointing out we don't like to talk bad about other products, because we think all of these products have some degree of performance, but we did avoid some ingredients like xylitol. And so recent studies have suggested a potential link between xylitol and heart issues. And so at least for the time being, until there's better data, and with working with the third-party toxicology firm that we work with, we're avoiding it, and for further investigation. And I think another really interesting example is nitric oxide, which it's a drug product. Technically in the US, it's an investigational drug, and it's really creating an active gas within your nasal cavity, which is known to have immunomodulatory properties, where there could be long-term side effects. And so right now, there's actually data out there that suggests that frequent use of NO can elevate your Th2 cytokines, which can mediate autoimmune issues. And the data that is available, as far as I know, is studying NO for like 7 to 10 days roughly as a post-exposure treatment. So very, very short periods of time. And again, it may work very well for post-exposure, but again, I think the point is there's just not a lot of safety data. And then the other, one of the really common questions that we get is, did you consider carrageenan in the development process? And the answer is yes, we did. We actually think it's a pretty interesting ingredient. It's obviously very common in other nasal sprays, but we decided not to use carrageenan, and we went with pectin instead. And we think pectin is superior to carrageenan, specifically for its efficacy against neutralizing influenza. And so if you look at, based on some of the data in the publication, pectin neutralizes influenza 99.99% in 10 minutes, which is pretty phenomenal. And it also acts as Nitin mentioned, as a physical barrier, which, as far as I know, has never really been reported in the literature. So it's kind of an really interesting discovery. And frankly, I think pectin is an underutilized antiviral ingredient. So I think that's all we have. I do want to provide a couple of product updates, and then we'll move to Q&A. And so, Kara, do we want to stop the recording now so we can go to updates?
Of course, my dog has to start barking immediately. Yes, we will stop the recording. I also will just remind everybody that we are now making a library of all of our still coveting recordings on YouTube. Alex, I'm just going to borrow your screen real quick and show people the YouTube channel so that they know where to find it. If you want to review afterwards some of the great information that Nitin or Alex has shared with us at Still Coveting on YouTube. You can also see our last Profi presentation, which had some of the similar data as well. So with that, I will stop the recording and then we can proceed.